Biotechnology is the application of living organisms or their parts to make technological advances and utilize those technologies in various fields. The modern era of biotechnology started with the discovery of the DNA structure as well as the development of genetic engineering techniques and the polymerase chain reaction. Biotechnology has many applications, especially in medicine and agriculture, including therapeutics, diagnostics, and genetically modified crops, etc. In India, biotechnology is entering a new era of growth as it is one of the key enablers for driving the bioeconomy of the country, which has grown to more than $137 billion in 2022 from $35.5 billion in 2014. Methods in biotechnology. The outstanding advances in biotechnology have been underpinned by the development and application of a range of biotechnological methods. Genetic engineering. Genetic engineering is the process of altering an organism's DNA using laboratory-based technologies. Recombinant DNA technology and the polymerase chain reaction are common methods of gene cloning. Recombinant DNA technology, the steps involved in the RDT are as follows. Identification and isolation of genes of interest from a host organism. Restriction endonuclease cuts the target sequence of these genes. Insertion of the gene of interest into a vector. Again, restriction endonuclease is used to cut the specific sequence in the vector, plasmids, that matches with the sequence of the gene of interest. Gene cloning. The recombinant DNA, plasmid plus foreign gene, is then transferred to a suitable host which is kept to grow on a nutrient medium. The host, bacteria, then expresses the gene, by producing proteins. PCR, unlike RDT, which is a cloning within a living cell, PCR replicates DNA in vitro, outside the cell. It amplifies DNA into millions to billions of copies in no time. It involves using short DNA fragments, artificial, called primers, to select a segment of the genome to be amplified. Then, TUC polymerase enzyme is used to amplify that segment in multiple cycles. PCR is used for the diagnosis of infectious diseases. RNA interference, RNAi. RNAi is a natural defense mechanism of plants and other eukaryotic cells in which small pieces of RNA shut down protein translation by binding with the messenger RNAs that code for those proteins. RNAi is specifically designed to silence or deactivate genes. Leveraging RNAi technology to silence genes has become useful for investigating gene functions, knocking down genes in diseases, and even developing pest-resistant plants. Tissue culture. Tissue culture is a biological research method that involves the growth of tissues or cells, plants or animals, in an artificial medium separate from the parent organism. In plant tissue culture, fragments of plant tissues are transferred to an artificial environment where they grow into a full plant, due to the property of totipotency in plants. Plant tissue culture has provided a way to propagate and genetically improve commercially valuable plants. It has been used to study characteristics of the growth, metabolism, reproduction, physiology, and nutritional necessities of plants under controlled circumstances. Tissue culture has permitted significant biological discoveries, including the identification of infections, enzyme deficiencies, and chromosomal anomalies, as well as the classification of cancers and the development and testing of medications and vaccines. Bioinformatics. Bioinformatics is a subfield of biology concerned with the acquisition, storage, analysis, and transmission of biological data. This interdisciplinary science harnesses mathematics, computer science, physics, and biology to develop software tools and methods to understand large and complex biological data. High-throughput data-generating experiments, including genomic sequence determinations and measurements of gene expression patterns, are used in bioinformatics. The potential applications of these biological data are in drug discovery and development. Applications of biotechnology. Biotechnology is applied in various fields at small as well as industrial scales for the betterment of humans. Some of these applications are medicinal biotechnology, Red biotechnology. Biotechnology is applied in healthcare for diagnosis as well as treatment of diseases. Gene therapy. It is a method that employs genetics to treat, prevent, or cure an illness. It works by replacing a defective or missing gene in a patient's cells with a healthier version. CAR T-cell therapy. CAR T-cell therapy is a kind of cancer immunotherapy treatment in which immune cells called T-cells are genetically modified in a lab so they can more effectively identify and eliminate cancer cells. Genome sequencing. It refers to sequencing the entire genome of an organism. Genomic sequencing is used for the diagnosis of diseases as well as identifying therapeutic targets of the diseases. For example, sequencing of the COVID-19 virus was extremely useful for the development of its vaccines. Therapeutic proteins. Therapeutic proteins are medicines that are genetically engineered versions of naturally occurring human proteins. These proteins are extensively used in the treatment of cancer, HIV, and other diseases. Examples: insulin, humulin, monoclonal antibodies, etc. Industrial biotechnology. White biotechnology. Biotechnological processing makes use of enzymes and microorganisms to create products that are useful in a variety of industries, to produce chemicals, materials, and fuels. The food and beverage industry uses S-cerevisiae, yeast. Microbial enzymes are used in washing detergents, food processing, cosmetics, etc. Energy. In the energy industry, biofuels sourced from plant materials such as corn stalks and grass are developed as renewable fuel alternatives to reduce the carbon footprints of fossil fuel production. For example, the two most common types of biofuels in use today are bioethanol and biodiesel. Agricultural biotechnology. Green biotechnology. Biotechnology gives farmers tools to make production more cost-effective and manageable. For example, genetically modified, GM, crops like Bt cotton have increased the productivity of crops. Environmental biotechnology. It uses biological processes to address environmental issues, including pollution control and renewable energy generation. Applications include genetic engineering, bioremediation, biopesticides, biosensors, and biofuel development, which aim to protect and restore the natural environment. Animal biotechnology. Biotechnology has helped to create many beneficial genetically modified animals. The first transgenic animal was the Dolly sheep. Biotechnology in India. The Indian biotechnology sector is one of the country's fastest growing sectors, and it is expected to play an important role in shaping the country's rapidly developing economy. Bioeconomy. It is the economic activity driven by research and innovation in biotechnology and life sciences, and enabled by technological advances in engineering as well as in information sciences. Potential. India's bioeconomy is on its way to achieving the $150 billion target by 2025 and over $300 billion by 2030. India is one of the top 12 biotechnology destinations in the world, and the third largest in Asia Pacific. Biotechnology is the technology of tomorrow and can leverage the wealth of India's bioresources, its unique biodiversity, its 7,500 km long coastline, and vast agricultural residues. Biotechnology is a trending career option among youth, due to increased attention to tools like genome editing, synthetic technology, microbial bioresources, enzyme and metabolic engineering, etc. Bio-startups. Because of the growing enabling ecosystem, the number of biotech startups in the country has increased from 50 to over 6,756 in the last 10 years. The government aims to increase the number of biotech startups to 10,000 plus by 2025. Subsectors of biotechnology in India. According to the revised India Bioeconomy Report 2023, India's bioeconomy sector stands at $137 billion, contributing 4% to the national GDP and employing over 2 million people.